As the host introduced, today we're going to have a discussion about the accelerated therapeutic antibody discovery through the ProSpeed single B-cell screening platform. For starter, I will give a really quick introduction regarding the technology background of the single B-cell screening approach. As of 2023, there are more than 100 antibody drugs that have been approved by the FDA. Most of them were generated by hybridomas. However, as a newly emerged technology, single B-cell approach contributed roughly 20% for the approved drug in the last 4 years. Single B-cell screening featured with relatively shorter timelines, better diversities, and better developability due to the concomitant pairing of the antibody-heavy and the light chains, but limited sensitivities and assay flexibilities. Alternatively, hybridomas, let me put up the laser. Hybridomas provides the most flexible screening scheme with the trade-off of turnaround time and diversities. Library display technology, on the other hand, is able to enrich for candidates bearing certain features like affinities, epitopes, but at the expense of diversities and developabilities due to non-concomitant pairing of antibody-heavy and light chain. Single B-cell screening is not just one platform, but comes with a variety of hardware to facilitate the process, such as the flow cytometry-based B-cell sortings, the microdroplets, microfluidics technologies. Among these different options, the Beacon platform achieved the best balance between throughput, accuracy, automations, sensitivities, and the assay flexibilities. Next, I will address the major bottleneck of the current single B-cell screening platforms and our corresponding solutions. To refresh everyone's memory, I will start with the overview of a Beacon workflow and the key features. Unlike hybridoma or phage library, single B-cell approach avoids complicated B-cell manipulations and takes a direct act on the screening of the B-cell at the B-cell levels. After plasma B-cell enrichment, the whole screening process takes place on a microchip within a couple of hours. Then those positive B-cells will be exported for cDNA recovery and sequencing, followed by recombinant antibody production and characterizations. The standard Beacon single B-cell workflow may look pretty straightforward, but given our experience with this hardware over the last 4 years, its intrinsic mechanism leads to several limitations. Due to the extremely low cell densities, the antibody enrichment level in the screening samples is not high enough for multiple rounds of binding assays, not to mention the cell-based function assays. Moreover, it also results in false positive and false negative due to limited signal windows and assay sensitivities. To solve these issues, people tend to sequence all positive B cells and make the recombinant antibody for further functional and binding confirmations. Imagine if you have hundreds of positive B cells, you will have to spend lots of resources on the part of sequencing, gene synthesis, plasmid preparations, and recombinant antibody productions. To address these series of bottlenecks, ProBiotic actually developed the ProSpeed expression system to facilitate the multiple runs of assays with accuracies, robustness, without the heavy cost of a molecular cloning work. The panel on the right is a typical workflow from positive B cells to validated hits. After the sequencing, the antibody sequence will be cloned into a expression vector, followed by transient expression and characterizations. This overall process takes about 5 to 6 weeks at least. However, in the ProSpeed expression system, instead of doing the sequencing and gene synthesis, the PCR fragments of the antibody heavy and the light chains will form a linear expression cassette to facilitate the transient expression. And after the binding and the functional validations, only those selected functional hits will be sequenced and purified. The overall timeline with this process is only 2 to 3 weeks, half of the typical workflow on the right panel. So unlike the typical workflow as I showed in previous slides, after exporting the positive B cells, we will do the antibody recombinant production, which is called the ProSpeed expression first, followed by the binding and functional characterization and sequencing. To put it in a simple word, we can do the antibody candidates profiling before recovering the sequence and preparing the plasmids. So with this upgraded process and workflow, we can get functional antibody candidates as fast as 1 month if you choose the express immunization, which only takes about 2 weeks. Comparing to hybridoma and phage library, the main advantage of this single B cell platform is the retaining of diversity by less manipulation of the B cell and earlier intervention of functional screenings. For instance, hybridoma will suffer multiple runs of diversity loss along the way. Only a very small fraction of a B cell can make its way to the functional screening stage. On the contrary, single B cell approach can carry a large percentage of a B cell repertoire to the functional validation step. To better illustrate the benefits of the ProSpeed process over the traditional workflow, I will share several application notes in the next couple of slides. The first scenario is to recover functional candidates from the positive single B cells. In this project, we identified 116 positive B cells using the Beacon platforms and produced the recombinant antibody supernatant for all of these 160 clones by the ProSpeed system without the sequencing and the gene synthesis. Then these recombinant supernatants were harvested for binding and functional validations. 85 of the 160 were confirmed as high binders, and 16 out of them were confirmed as functional blocker by the IL-8 reporter assays. The cost of this work is about 42,000 US dollars, while if you choose the traditional workflow, the cost is going to roughly double at 90,000 US dollars. The second scenario is to obtain specific binders against a matrix of positive and negative antigens. As I mentioned at the very beginning, the Beacon platform can run the on-chip screening for up to 3 antigens before the B cells start dying. In this project, 156 positive B cells were exported for the ProSpeed linear expression. 56 and 46 of them were confirmed as ELISA and phage high binders, and 14 out of them were confirmed as candidates with minimal cross-reactivity to 3 irrelevant targets, the counter-screen antigen 1, 2, and 3. These 3 irrelevant antigens share high homology with the positive antigens. The cost of this work is about 47,000 US dollars, while if to choose with the traditional workflow, the cost will also be doubled at more than 100,000 US dollars. The third scenario is to improve the assay sensitivities to reduce the false negative selections. In this project, we identified 86 positive B cells, in which 38 of them were potential F1 cross binders. F1 is actually a family protein of the target antigens. After the ProSpeed linear expression and binding confirmations, 10 of them were actual cross binder to the F1 antigens. The overall cost is roughly 20,000 US dollars, and it may be tripled if we choose the traditional workflow. The fourth scenario is to improve the assay sensitivities to identify the true cell binders, especially when you run the assays using the cancer cell lines instead of the engineered cell lines, because cancer cell lines usually comes with relatively low antigen enrichment level on the cell surface. In this project, 121 positive B cells were exported at the B cell screening by the screening against the cancer cell lines, and 24 of them were confirmed as high binders, high cell binders against the cancer cell line. The last scenario is to rescue those missed hits due to the unsuccessful molecular cloning work. In the traditional single B cell screening process, the overall success of the screening process heavily relies on the success of the PCR and the recovery of the PCR fragments. Since A single B cell can only generate tiny amounts of mRNA. The PCR reaction may not generate enough amount of cDNA, and the trace amount of cDNA is not able to support the sequencing process. However, this trace amount of cDNA can still be incorporated into the linear expression cassette, the ProSpeed system, for antibody production and the follow-up sequencing. As a result, these non-survival hits can be recovered by the recombinant production and molecular sequencing. In summary, this upgraded workflow offers a comprehensive solution to solve the bottleneck of a current beacon-based single B-cell platform, including feasibilities of multiple binding assays, functional validations, improved sensitivities, and the potential rescue of lost hits. Moreover, the feature of the expedited timeline and higher diversities coupled with our experience over 100 projects makes it a favorable solution for antibody discovery campaigns. For the last part, I will share two case studies of using this ProSpeed single B-cell based system. One is the IgG discovery against the DLL3, and the other is VHH, the single domain antibody discovery against HSA proteins. This illustrates the general workflow of the IgG discovery against a popular cancer TAA target, the DLL3. It's pretty much the same as we have discussed in the previous slides, so I will not get into the detail of this overall process. The first part of this project is antigen validation and on-chip binding assay development. The purpose of this work is to make sure that the antigens gives a decent signal windows. It is a crucial step to verify these details before carrying out the screening process. Once the single B-cell screening is initiated, there is no room for further troubleshooting. After immunization, both mice strains, the biopsy and the AJ, demonstrated decent serum titers across human, cyno, and mouse antigens. The B cells from those selected animals were split equally into two halves for both beacon single B cell screening and hybridoma based screening. Those B cells were screened sequentially by human cyno and mouse DLL3 recombinant protein and human DLL3 stable cell line. These recombinant protein antigens were biotinylated and locally enriched by streptavidin conjugated beads. Then the antigen-specific antibodies and the fluorescent secondary antibodies were captured sequentially onto these beads for detections. The cell line antigens is detected in a similar way by primary antibody secreted by the single B cell and fluorescent secondary antibodies. By doing this sequential binding screening, we obtain 4-panel uh, positive B cells against each antigens. And based on the overlapping regions, we can easily identify candidates fit for different screening criteria. These positive B cells were exported and processed through both the uh, the ProSpeed expression system and the traditional single B cell workflow simultaneously. The recombinant supernatant demonstrated decent antibody concentrations ranging at microgram per milliliter levels on the figure 1 at the top right panels. It further proved that given this antibody concentration in the supernatants, it can generate reliable screening results using the supernatants instead of the purified material. Besides both workflow, either the linear expression cassettes of the ProSpeed or the traditional circular plasmids uh, gives consistent binding profile as shown in the figure 2 on the lower right panel. We did further uh, affinity characterization with cones generated from the big single B cell approach on the top panel and the hybridoma approach on the bottom panels by ELISA EC50 and against human cyno mouse antigen and flow cytometry on the right panel against engineered overexpression cells and the DMS53 cancer cell line. Candidates from the single B cell approach on the top showed a relatively more diverse binding profile affinity range uh, than those from the hybridoma at the lower panel. In addition to the affinities, we also did epitope analysis by competitive flow cytometries against two clinical benchmarks, the BMK1 and the BMK2. The candidates from the single B cell approach in the top panels also demonstrated a more epitope coverage comparing to those from hybridoma approach on the lower panel. To further analyze the candidate diversities, we plotted the germline coverage with a heat map on the left panel to compare these two different approaches, the single B cell approach versus the hybridoma approach. Candidates from the Beaker single B cell screening clearly shows a much wider distribution of germline. The phylogenetic analysis on the right panel further demonstrated a more scattered patterns for the sequence homology analysis, as shown uh, as the green one. The green one is generated from the single B cell, while the black one is from the hybridomas. So that's the story for the first case study. In the second case study for the single domain, the VHH discoveries, the overall workflow is a little bit different from what we have discussed so far. Due to the lack of alpaca plasma B cell enrichment kit, we adopted a different strategy as illustrated here. After immunization, the alpaca memory B cell, instead of the plasma B cell, were enriched by antigen proteins, followed by incubation in vitro uh, for maturation of these memory B cell into plasma B cell. Then these processed plasma B cell were loaded on the chip for beacon single B cell screening, the ProSpeed linear expression and validations, the same way as we previously introduced. During the immunization stage, serum samples were tested against human cyno and mouse HSA recombinant protein. In addition to the total IgG titer as indicated by the blue curves, the IgG2 and IgG3, which is a representative of the VHH or the single domain titers, is also included as a crucial evaluation of the immune response. So the IgG2 and the 3 are indicated as the purple curves. The VHH titer is actually a key indicator for us to anticipate the VHH hit rate in the downstream screening process. This is a high-level summary of the screening results. We recovered roughly 150 positive B cells from each round of the PBMC samples after the third and the fourth immunizations. Taking the fourth immunization as an example, among 153 positive B cells, 95 and 95 VHH and 21 IgG were successfully cloned or PCR. We only selected those VHH PCR fragments for sequencing, and 92 out of 95 were successfully sequenced, representing 84 unique sequences. After recombinant antibody production, 75 out of 84 were confirmed as actual binders. These numbers represent roughly 80% of conversion rate from positive B cells to confirmed binders. We did further sequence alignment for diversity analysis with candidates generated from two batches of the PBMC, the one after the third immunization and the one after fourth immunizations. Unique sequences identified from these two different batches evenly distributed to overlapping sequence clusters, demonstrating a decent sequence diversity and coverage. However, only one sequence were identified in both PBMC samples, suggesting that the B cell pool in the immune system, from the immune system of the animals, is still under high frequency of somatic hypermutations during the immunization stage. At the end, I would like to say a few words about ProBio ourselves. We have been in the discovery CRO business for 2 decades, and at least 15 pipelines delivered are in the global clinical stage. Our end-to-end platform aims to accelerate the drug discovery process to facilitate the development of a variety of drug modalities, including monoclonal or bispecific antibodies, ADCs, and cell therapies. Thank you everyone for your time. I think that's the end of my presentation. Let's move on to the Q&A session.
Thanks for Dr. Derek Chen's excellent presentation. Now, if you have any questions, please type it on the chat or Q&A panel. As you enter questions, please also follow our LinkedIn account, Quick Profile. And the Korean audience can also scan this QR code and follow our Kakao channel. There will be more online sharing and resources, and we will share them on our LinkedIn and the Kakao channel at the first time. If anybody have any questions, if you have, you can type it on the chat or Q&A panel. Oh, okay, we received some questions from the audience. Let's look at them one by one. The first question, what will happen if we do more than 3 rounds of screening on the single B-cell chip? Dr. Chen?
Could you speak a little bit louder? I can barely hear your questions.
Okay, what will happen if we do more than 3 rounds of screening on the single B-cell chip?
Okay, again, so what's gonna happen if we do more than 3 rounds of screening on the chip, right? Yes. So as I mentioned in the during the presentation, normally we do not recommend to do more than 3 rounds because it takes more time, and the longer the B cells stay on the chip, there's a higher chance that the B cell will start dying. And once the B cells start dying, even if we identify as the positive B cells, we will not be able to recover the intact sequence because the mRNA will start to degrade, and we will not be able to get a successful PCR fragment with the full length of the rival regions, which will make it futile for the whole process of screening. Even if you get a positive B cells, you cannot recover the sequence.
Okay, thank you, Dr. Chen. And the next question is, how did you simulate memory B cell to plasma cells in ALPACA?
So let me just confirm. The question is, how do we stimulate the memory B cell into the plasma B cell, right?
Yeah.
Yes. So I think there are a bunch of published papers describing the general process. Basically, we use the antigens and a series of chemicals to induce or to activate the memory B cells and to lead the maturation from the plasma B cell to from the memory B cell to the plasma B cells.
Okay, and the next question is, can you run functional assay on the single B cell chip, or can it be done using the ProSpeed expression reagent?
Can you repeat that question? I can barely hear you.
Okay, can you run functional assay on the single B cell chip, or can it be done using the ProSpeed expression reagent?
Okay, so the question is about cell-based function assays. For the first part, the answer is no. As of now, we haven't been able to establish the cell-based function assays on the chip levels. So that's why we developed this ProSpeed, the linear expression system, as a alternative solutions. Instead of running the assay on chip or doing the whole gene synthesis and the recombinant antibody expression and the purification and use the purified material to run the assays, we developed something in between that we can clone variable region out of the single B cells and use the PCR fragments as the plasmid. You do not have to generate circular plasmid. It's just a linear version. Just use this linear version of a PCR fragment to do the transient transfection in the high throughput and small volume fashions like 1 milliliter systems. And since there's only 1 milliliter volume for the transient transfections, there's no need to do the purification. You can just harvest the supernatant and run the binding and the cell-based function assays. The typical range of antibody concentration in the supernatant is from a single digit microgram per milliliter to like even 100 microgram per milliliters, which is more than enough for the majority of binding assays and cell-based function assays.
Thank you, Dr. Chen. And there's a last question. What is the advantage of using single B-cell approach over phage library for VHH antibody discovery?
Okay, so the question is, what's the advantage of using the single B-cell approach to do the VHH discovery comparing to the library? So first of all, library is probably one of the most established and popular approach to do the VHH discoveries. Especially VHH only comes with just one domains, so there is no concerns about heavy chain and the light chain mispairing uh, with the phage libraries. However, as I shown in the presentations, uh, the single visa approach definitely offered additional benefits or features. Things like, first of all, we can see or save a lot of time, especially the time you spend on the library constructions and the library panning and repeated screenings. So basically, once the immunization is completed, you only need 1 day to do the screening and roughly 1 to 2 weeks for uh, the linear expression and the binding confirmations. You can get confirmed binders. Uh, I mean, the overall timeline from the immunization to the confirmed binders using the single B cell approach is a couple weeks shorter than the phage library. That's the first features. The second feature is, of course, the diversities. When you use the phage library to do the antibody discoveries, there is significant amount of loss of diversities, and you cannot even control the loss of diversities because during the panning stage, uh, you do not know which part of the sequence or which part of the B cell pool is discarded or which part of the B cell is retained. You cannot control that. That's the major source of diversity loss for the phage. However, using the single B cell approach, in theory, every single B cell that is able to secrete the VHH will be carried over for the antibody screenings. So that's why it can provide significantly better diversities based on the process itself and also our data of the sequence analysis.
Thank you, Dr. Chen, for answering the advantage of using a single B-cell approach. There is another question. And now this how will
I'm sorry, I cannot hear you.
and nanobot allow kind of screening? How will B-cell
Okay, I can take the question from here. This is a question we received from the pre-webinar registration. The first question is, can nanobodies allow this kind of screening?
If the question is whether we can run nanobody discovery or nanobody screening with the beacon, the answer is yes. We showed a case study for the last part of the presentations, which is a VHH or nanobody discovery against the HSA protein using this exact the beacon single B cell system.
Okay, that's great. Yeah, and the second question is, how long does the B cell platform cost?
Well, actually, the price range for different projects is the price covers a pretty wide range of the price depending on the project itself. Typically, well, it's maybe 30 to 80,000 US dollars, depending on uh, the nature of the targets, the scale of the screening. We can provide more detailed pricing and technical introductions after the meeting based on the further information reviewed by you and your team.
Okay, yeah, thank you. That's all the questions from my part. Hi, Xiaojing, you can go ahead. Hello, Xiaojing, can you hear me?
Can you hear me?
Okay, I can hear you now.
That's all of our questions today. And on A40, if you have any in this webinar, you can.
Yeah, maybe we cannot hear you very clearly. Yeah, I think that's all the questions we have for today. And we are also going to hold another webinar, Cost Reduction Strategies in Clinical Stage Process Optimization for Biologics, on 14th May. If you are interested in this topic, please register this webinar on our website. I will also share the link to the chat room. Okay, if you are interested in this webinar, you can find the link in the chat room and register it. Okay, thank you all for attending today's webinar. And if you have any questions, you can contact us through our official email. And let me join others in thanking Dr. Duck Chen for his time today. Look forward to meeting you again soon. Thank you. Bye-bye.
Thank you. Have a good time. Bye-bye.
Thank you. Bye.